Welcome, everyone, and thanks for joining us this morning. My name is Tom Lynch. I'm the president and director of the Fred Hutch and the Raisbeck Endowed Chair. It is really fantastic to be with all of you this morning. You are people who have made such a difference to the Fred Hutch in supporting this incredibly important antiviral research. And again, I thank you. And I thank you for your interest. I thank you for your support. And we're really excited to be able to talk to you today about where things stand in our research in herpes virus. I want to start, as we do all of our public meetings at the Fred Hutch, with a land and justice acknowledgement. We start all our events by sharing this, recognizing the land that we're on and the importance of that land to the people who are here. The pandemic has spotlighted how communities of color are disproportionately affected by disease, and our mission is to reduce suffering from cancer and other diseases like herpes simplex virus for all people. We acknowledge that we work and live in the traditional lands of the Duwamish, Tulalip, Muckleshoot, and Suquamish tribal nations. We thank the original caretakers of this land who are still here. We support the ongoing struggle for justice against racist, religious, sexist, xenophobic, ableistic, trans-antagonistic, and all oppressive violence. We recognize with gratitude those who sacrifice, struggle, and labor to make our freedoms possible and challenge us to learn, work, and live justly. So again, recognizing that is important at the Fred Hutch and other places as well. So I want to thank you for joining Keith Jerome and I today to talk about herpes simplex virus, or HSV research. We know that this research means a tremendous amount to people whose lives have been permanently altered by HSV. And in my discussions with Keith Jerome before we had this event, Keith told me what an incredible group of people you are, that you are passionate about the research that he's doing, that it's an incredibly informed group and dedicated to finding a cure for this condition. You're also great fundraisers. You've raised more than $180,000 for HSV research, and the supporters come from 50 states and 34 countries, and you make up almost a third of our monthly donors to the Fred Hutch. So again, I thank you for your help and for seeing how important this is. I think this is a year where we probably don't need to remind everyone how important viruses are. But they are important, and folks like you who've been supporting this work have recognized how critical viruses are in our society for quite some time. And we know that herpes simplex viruses affect billions of people around the globe, and yet this research is chronically unfunded, and you're making our search for cure possible. I want to say a couple things about the Fred Hutch before we get going. Our mantra at the Fred Hutch and the way we think about our work is that cures start here. We've developed cures for cancer, and we're dedicated to finding cures for diseases like COVID, HIV, HSV. For those of you, about 30% of our research is antiviral research at the Fred Hutch. And again, very timely this year, but we're committed to viruses whether COVID's here or not here, as you know. We're committed to HSV in the work that Keith's doing, as well as a large effort in HIV and other viruses. Now, you might wonder, why would the Cancer Center be so interested in viruses? And it's really for two big reasons. The first is, when we started as a cancer center, we weren't like covering all cancers. We were basically a place that focused on bone marrow transplant. In fact, bone marrow transplant was discovered and developed at the Fred Hutch. And what they found was many of the patients who had bone marrow transplants were developing severe viral infections, and they wanted to understand those viral infections better. And that's why the first efforts were made to get into virology. And I think a second big reason for why we're in virology is that viruses cause cancer. And so viruses cause more than about 25 to 30% of all human cancer on Earth are caused by viruses, many of them herpes viruses. And obviously we can talk to Keith about the role that herpes viruses, not necessarily HSV, but other herpes viruses can play in cancer or initiation, since that can happen. The Hutch is synonymous, and our approach now is talking about the importance of fearless science, We believe that this is incredibly important. We believe it's important for our scientists to be enabled to take our scientists to be enabled to pursue ideas that are likely to make a big impact. And that's one of the things we're really pushing at the Fred Hutch. And I think you're seeing that whether we're talking about Keith's work in HIV, whether you're talking about what we're doing in COVID. For those of you who may or may not know, the Fred Hutch is a major organizing site for designing and understanding the COVID vaccine trials. So tremendous amount of work on COVID vaccines have been done here. One of the questions I look forward to asking Dr. Jerome is, why has herpes virus been such a tough target for vaccination? And I really look forward to learning from him as well today. And so that is a great way to introduce Dr. Keith Jerome. And he's even more thankful for your efforts than I am, because most of them benefit his laboratory that's doing incredible work. Keith is a renowned virologist whose research focuses on viruses that persist in their hosts, such as herpes simplex, HIV, and hepatitis B. Many of you who are in the Seattle area will know that Keith is also the head of the viral testing lab at the University of Washington. And he probably has become more of a coronavirus expert than he ever imagined he would be. In fact, when you look at the classes of viruses, to think that coronavirus would become the biggest thing in his life right now is probably something he might not have been able to predict. But Keith plays a huge role in the testing effort. His laboratory at the University of Washington is it's the best testing lab in the country for viruses, and it's been at the forefront of how we can test for viruses. And so that ability was incredibly important that he's done. He's tested more than 1 million people, which is really fantastic. And then this is at the same time that Keith and his colleague, Dr. Martine Aubert, were finishing up their successful study using gene editing to eliminate HSV in mice. So, it's not like he gave up his day job. So he was working in developing this incredible viral testing lab while still being completely committed to HSV. And that's one of the things I'm incredibly proud of at the Hutch, is that so many of our people who were working on HIV didn't stop working on HIV. Our cancer people didn't stop working on cancer. They did that and they worked on COVID. So look forward to learning about the gene editing strategy today and hear where we are. So Keith, let's start with asking you to tell us a little bit about your paper that was published in Nature Communications. Tell us a little bit about the findings. What does it mean, and what's your perspective on it?
Sure. Thanks, Tom. Thanks for the nice introduction. But before I even answer your question, I'm just so thrilled to have this group on the line to be able to say thank you in person. I've actually thanked you guys in many of the talks that I've given. It's typical in science that kind of you get to the end and you say who funded your work, and typically the scientists say, oh, it's NIH did this, or some rich person did this, but it's really fun to say this many hundred people, and this, we don't name everybody by name, but just this grassroots effort is actually making this research possible. So I truly thank you, and I think that there's so many people who've been affected by these viruses who don't even know you're doing this, and this is part of the unsung nature of science, but I know that you would have their gratitude as well. So I wanted to just say that right up front. Tom, I was incredibly excited about that paper for a couple of reasons. First of all, that's a long time goal of mine, of our lab, is to really be able to do something about these latent HSV infections. And I've said this many times in talking with different groups. When we started this work, even to say, I'm a scientist at a well-known institution, a renowned institution, and I work on a cure for herpes simplex, it was sort of like people think you're a quack. What are you talking about? Because we've seen all this stuff on the internet, and so it was really exciting to publish that. First of all, to get the results, but then to publish that paper in a really respected journal where it got a lot of attention in the lay press and other places. I hope that it catalyzes work in this field. That we're not the only group working on this. I hope that other groups get interested and we actually get the body of science. You've seen in the COVID pandemic how much the scientific enterprise can actually achieve when it's focused. And I'd love to see some more focus on this virus. So the basic finding of the study is we can latently infect mice with herpes simplex. We put the virus on the mouse and it establishes an infection, a lot like human beings, that it goes down into the neurons of the ganglia. It basically goes to sleep just like it would in a human being. It expresses the same genes it would in a human being. The only difference is it doesn't reactivate like it does in a human spontaneously. So we don't have that part of it, but the virus is there. And then we can take this gene editing approach, which is sort of molecular scissors. The virus, when it's latent, lives in a little circle in these neurons. Those are the nerve cells, bodies of the nerves. Virus lives in there. We basically make two cuts in those, and it basically breaks that circle into two parts. And that virus can't repair itself. Once that happens and the body degrades it, it goes away. And basically, we were able to show, we'd shown that process in the culture dish for a long time. And other groups have shown that. We've shown it for some other viruses. Other groups have shown it for other viruses. But this is the first time for any virus that process has been shown in a living animal, in a living organism. And the way I look at it, if we can do this in a mouse, well, gosh, then, you can start to believe you can do this in a human being. So I see this as a pathway toward our ultimate goal, which is the cure of this infection.
So Keith, tell us a little bit about exactly how you do this. What's the technology used to edit this gene out?
Yeah, so you simplify it a lot. You say, you just go in and you make these two cuts with these scissors. It sounds pretty simple. It's really not. The technology, the scissors, were sort of a precursor, an earlier technology that's been in large part supplanted by CRISPR-Cas. So maybe a lot of people have probably heard of that, this gene editing, cutting stuff. This is an older technology, or maybe a way to think of it is earlier discovered technology to do that. It's been around for maybe 15 years or so. And basically, what these things do, if you remember that, all of our DNA and our cells are in a virus. It's made of these four letters, right? A, C, T, and G. And what these things do is that they just start reading the entire way through, and they look for a very specific sequence of a 20-letter-long word, if you will, within those hundreds of thousands, or for humans, billions of base pairs. And if they see that, and only if they see that, they make a cut there. So that's when the scissors activate themselves. And the scientific name for these scissors are called meganucleases. And they're in nature, they're found in yeast. They recognize these long sequences. The technology that's really been developed largely by my colleague, Barry Stoddard, here at the Hutch, is to find these things in nature, find the 20 base pairs that they're, the word they're looking for, and then change those 20 letters, change that word into the letters of the word of something we want to cut, in this case, herpes simplex. So then that's how we get that specific cut in herpes while we leave the host genome alone. Okay, so there's the scissors. How do you get them where they need to go, right? And this is, in some ways, actually the bigger challenge. And so the technology we've been using is actually something called Adeno-Associated virus vectors. Adeno-Associated virus in nature is something that pretty much all of us have been infected by. We don't even know it. It's something called a defective virus, that it only works when it has to piggyback along with another virus, and it sort of uses parts of those other viruses to replicate itself. It doesn't cause any disease. And so it only has two little genes in it. And what we do is we take those two genes out and we plug in the coding sequence for these scissors. Okay, so we plug in the DNA that will actually allow the virus to take this to wherever it goes and produce the scissors within the body. And so, gosh, I don't know how to convey this, but the months and really years of hard work going through all the different flavors of AAV that exist out there that have been discovered over the years or developed to use as vectors for different sorts of gene therapy. And we just went through dozens of them until we finally found a few that really worked and figured out how's the best way to put them. Originally, we sort of put them into the skin of the mice, just under the skin. Eventually, we found just injecting them into the blood worked even better. And so that's where we are now. But you see the end product, you see this breakthrough. What people don't necessarily see is all the painstaking experiments and the failed experiments that were sort of behind the scenes that allowed us to get to that point.
Great. So Keith, one of our primary objectives today is to answer the questions from the folks, and that's really why we're here, guys, is to answer your questions. And so what we have is we have the top 5 questions that you folks shared ahead of time. So we asked for questions in advance, and these are the top 5 questions. We're going to answer these first, and then I encourage you all to use the chat line and put more questions in there, which I think will probably come up, particularly as we answer these questions. So Keith, you're probably not surprised that the first question is the first question I would have as well, which is, as a clinician, I'm a clinical doctor, a clinical lung cancer doctor. I hear about a great, exciting thing. I want to see, can you do it in patients? I mean, look at COVID. I mean, they were able to, the first vaccine followed the first sequencing by about 3 weeks in terms of how they were able to do that. So what are the obstacles? If you can do it in a mouse, what are the obstacles to trials in patients? And do you think that the AAVs, which you used in this, will be effective in humans? Can you assume because they work in mice that they work in humans?
Yeah, so those are great questions, and I get those a lot. And Tom, the scientists always hate saying when something's going to happen, because the nature of science is we're pushing the unknown. So things come up that we don't anticipate, and there are obstacles we have to fight through. So it's always hard to say. If you ask me how long will it take to cut my grass, I know. If you ask me how long it will take to write a 300-page letter, I know. To achieve a scientific goal, I can give you some guesses. And we've said we're at least a couple of years away from a trial. There's a lot that needs to happen, and the single biggest one on our mind now is making sure this is safe. The last thing we want to do is hurt anyone, and it'll be brave people who do this the first time. We have to be very honest, and there's a lot of motivation, and we've talked with folks, and their people are willing to be involved in trials, and there's a tremendous altruism here that's really inspiring. But we need to make sure that these things are safe, and so we're looking at our animals, trying to understand exactly what's the right dosage of the AAV and the scissors to use. And we're just beginning now to really look not just scientifically, is it safe, but actually now starting to think about, when you go through the regulatory process, you go through FDA, who will have to approve this work, what exactly are the milestones of safety that they're going to want to see? And so, that's a series of conversations that really will happen over the next couple of years now to get to that point. One of the challenges that we face, Tom, compared to COVID or cancer, I hear this a lot. People don't die of HSV. That's generally true. There is, HSV encephalitis. A lot of people have heard of it. It's extremely rare, but it can be fatal. So there is a burden of safety that's very, the bar is set very high for us. You have to really prove that. We've learned a lot. And this will probably come up several times today. This is not the only virus I work on. I work on HIV. I work on hepatitis B. We talked about COVID. This is probably the one, that I know the most about. Frankly, it's the project that's farthest along in the cure. So, this group, you're number one, which is great. But one thing I like to talk about is how much we learn from one virus to another. When we work on HIV, part of the breakthrough of, making those 2 cuts, I'll just say, really came out of the HIV world. And maybe I can explain later how that insight happened, that you make 1 cut, it's not so good, the virus actually fixes itself. If you make 2, it doesn't know how to do that. It actually falls apart and gets degraded. And that was sounds simple in retrospect, but it was an incredible insight that we had to have to move forward. So one thing we've really learned from the HIV world, where the cure agenda is more mainstream. There's a lot of NIH support for an HIV cure, and we've had these discussions because HIV now is in a developed country is a non-lethal infection. We have good therapies; people can live normal lives on antiretroviral therapy. So there is a burden of safety, and a lot of those concerns have been worked through there. So we're able actually to sort of piggyback on that. And fortunately, through my direct involvement in that, kind of know what some of those are. So we sort of know who to talk to and what needs to happen. It's going to speed this work up.
Yeah. And so I guess one question, Keith, that I would have is whether you're using a meganuclease or using CRISPR. I think the thing we worry about is, making those two cuts is great if it's just in the viral genome. How do we know that it's not going to make cuts anywhere else in the genome? Tell me a little bit about the specificity. And I know with some of the early CRISPR trials, there have been some unexpected outcomes, also some great benefits so far. So tell me a little bit about how, when you think about any kind of gene therapy, how do you know that you're specific enough?
Yeah, that's a really important question. One thing that we look at is, first of all, how did the animals do? And, for some of them, we leave this in for months and months, and they seem to do well. So the animals seem like they're fine. Now, that's not perfect proof. Of course, humans live a lot longer and, are bigger, and there's so many things, and they're just different. So it's not perfect proof, but it certainly means there's every reason to think this could be safe. So one of the really important questions is this idea of off-target cutting, that, you're cutting DNA, you want to cut the virus, but what if you have a human genomic sequence? There's 3 billion base pairs of a human. The virus is only 150,000. Human is 3 billion, okay? So how do you know there's not something in there that looks kind of like the virus? Well, we basically look at that right at the beginning of this process through computer databases and make sure there's nothing that's close in a human being before we want to target something in a virus. It has to be very different from any human sequence. We know how close they need to be for the scissors to work at levels that we can typically measure. And then what we do is that, when you do that, you still have the most closely related sites. So let's say if I tell you that the meganucleus needs a 20-letter long word to be exact, you might find that in the human genome there might be a site that shares 15 of those 20 letters or 14 of those 20 letters. We identify the ones that are the closest. And then in the treated animals, we actually take those sequences and we look through this, what we call next generation sequencing. It's just, we literally take hundreds of thousands of cells and pieces of DNA, and we just sequence them over and over and over to see whether any are mutated. Is there any evidence that there's been any off-target cutting? And the answer is, when we do that, we haven't seen that. So that tells us that if there is some off-target thing, it's very low. We're working on additional technologies now to try to do a very unbiased look. There we have to sort of guess where the off-target sites might be. We do that through mathematical approaches and computers, predictions. We're actually developing approaches where we can look in a non-targeted way, just at the whole genome, and make sure that we really can't see any problems. Particularly in the CRISPR-Cas field, there are many, many labs working on increasing the specificity of those scissors. Yeah. So that's an area of very active research, and we piggyback on all that research. I talk with those people, I go to meetings with them, I read all their papers. With the meganucleases, it's a smaller field doing that. We believe that if you sort of the key, I don't know how to put this exactly, but the match between the scissors and the target is physically just bigger and more demanding for a meganuclease than it is for CRISPR-Cas9. So we think that they're inherently probably more specific, but it's something that we need to look at very carefully for those, because there are fewer people in the global scientific community looking at the meganucleases and their specificity.
So Keith, one of the 5 top questions is actually that specific question, which is, at the Hutch, we have several CRISPR-based researchers who are looking at CRISPR-based gene therapies for correcting diseases like sickle cell anemia and other diseases like that. And you have a chance to work with those colleagues and learn from them. So any thought that CRISPR-based techniques could turn out to be a way to go after HSV? You talk about how hard the meganucleases are to work with. What about CRISPR? Do you think there could be CRISPR-based gene editing that you could, now that you've proven the concept works, could this be applicable to a CRISPR-based system?
Yeah, so actually, if you go deep into the back kind of half of that paper that we published in Nature Communications, we actually tried to do this with CRISPR-Cas, and we could get a little bit of gene editing of the virus, but we couldn't get anywhere near the levels. So we could sort of see that we had made a few cuts in the virus, but it wasn't enough to actually make it degrade. So we didn't get that loss of the virus in the animals. I don't fully understand why that is. I'll be really honest. We have a couple of hypotheses that we haven't been able to completely tell apart yet. Probably our leading hypothesis is that if you just look at the relative size of the scissors, the meganucleases are sort of, a kindergartner's scissors, and and Cas9 is kind of garden shears. I'm exaggerating a little on the difference, but one is kind of small and one is good. And it's sort of like, well, if you just need to nick a piece of paper, maybe you don't need garden shears. And I don't know if I can keep pushing this metaphor much farther, but I talked about, that we deliver these with AAV, and because the meganucleases, despite the name with mega in them, they should be mini nucleases actually, because they're tiny. They can fit really well into that vector, and then we can do a bunch of genetic tricks that I won't talk about too much, just smart stuff that molecular biologists in my lab have figured out, so that you get really good expression of that once it gets to the cells. So we get a lot of the enzyme where it needs to be. CRISPR-Cas9 is so big that it just barely fits into the vector, and so we can't quite optimize it quite as much as we'd like. You just don't have the room to make those little tweaks. And it may be as simple as that. There's other things that are a little deeper. Meganucleases actually evolved in yeast, and yeast sort of put they decorate their DNA almost like a Christmas wreath with things coming off of it, whereas CRISPR-Cas9 evolved in bacteria, and they don't put those decorations on. So it's possible that our DNA, the viral DNA in us, is decorated. So maybe the meganuclease is actually better at sort of seeing past all the glitter and getting to what they need to do. I'm working with a lot of our really basic science colleagues. They're fascinated with that idea. We call it chromatinization. That's the fancy word for decoration. But basically, it could be that in us, our viral DNA is just protected, and you need something that's kind of evolved over billions of years to get past all that and do its work.
And I'm not trying to imply that CRISPR is there yet. I mean, it's you and I.
Yeah.
There's a bunch of companies working on CRISPR and investigators working on CRISPR-based approaches. There's huge obstacles we have to overcome with CRISPR as well. I didn't mean to imply that CRISPR was going to be easier.
No. And yeah. Tom, I'll just add, because you forgot something you want to say. I'll say we've not given up on CRISPR-Cas9 at all. And in fact, we're collaborating with some of the other researchers groups who are working on that. And I've told them, maybe we just didn't do it. Maybe there's something we're missing. Maybe we didn't optimize it right. Maybe you can help us. And we're going to try that again, because there are some really nice advantages of CRISPR-Cas9, including in a very practical way that there will be other human trials where it gets put into people for other diseases. And if those exist for Cas9, it may make FDA more likely to approve our work, whereas we would probably be the first people to put a meganuclease in a human trial, and there'll be added scrutiny. So if we can get CRISPR-Cas9 to work, there could be some real advantages.
So another question, the third question on the list from the audience, which is a really important one as well, which is obviously many of the people on our call are people who have HSV, and I remember when I was in practice, one of the areas where my patients were most concerned about was the idea of shedding and outbreaks being the big concern. And so one of the questions that's been asked is, do you expect that the reduction in shedding outbreaks from this experimental treatment will be lasting? If not, are there any potential workarounds that could make this possibly be more lasting? And the other question that's part of this is there a possibility that the virus might be able to reinfect patients after this kind of a treatment?
Yeah, those are both great questions. So the way that we put the scissors in now with AAV, AAV is actually really stable in cells. Once it goes in, it just hangs out, kind of makes the genes that we put into it. And the way that it goes away in a lot of tissues is that the cells divide, and as they do, the AAV doesn't divide. It just kind of, part of it goes to this daughter cell and part goes, this gets washed out over time. Our neurons don't divide, so that once the AAV goes in there, it's there. And so we've seen in our animals that the expression goes on really, at least for the life of the animals. So this would be at least for years in human beings. What that means is those cells are protected. We think we're actually doing experiments right now to demonstrate this in a very formal way. But it looks like basically not only do they actually eliminate virus once it exists there, but they destroy any viruses trying to come in. Makes complete sense. We just need to demonstrate this formally. So yeah, you'll get this long-term protection. One of the interesting things that we've really thought about in the herpes virus field, it's actually interesting that there are fundamental things we still don't fully understand, and one is to what degree the virus in neurons actually sort of replenishes itself over time. As people have outbreaks, the virus goes out to the skin or mucosa along the nerve axon, and then the virus replicates out there. And then how much of it comes back, and does that recede any of the nerve bodies again back in the ganglia? And so, some of the people in the scientific field are frankly most excited about our work because it's sort of a probe to answer these scientific questions they want to do. And we're happy to work with those folks. I tend to be a little bit results oriented, like we're trying to actually help people. That's part of what the center does, as you well know. But if we can learn some things that probably have useful benefits in other places, so happy to work with them. But we're really trying to understand those things, and this question of reinfection is important because one thing we're looking at, Tom, is I talked a lot about AAV. We're also interested in other ways that we might deliver these. So if we could come up with a completely there's things that we call nanoparticles that are actually just produced in a laboratory, sort of in a, almost think like in a, if you're producing them at scale, it'd almost be like in a beer fermenter sort of thing. Like in fact, some pharmaceutical companies actually started out as making alcohol, and then they moved over into doing these other things. So you can make these in large amounts, and they wouldn't be a virus, which again might be a little bit more acceptable to FDA. We'll see about that. But using those sorts of approaches, the expression of the nuclease wouldn't necessarily be lifelong. So we really want to understand how much reseeding of the reservoir of the neurons there really is to understand, would we need to treat people a couple of times if we did it that way? Or is, if they just get expressed for a month and then go away, is that enough? Those are questions we'll answer over the next couple of years. But these are the sort of things that just, are the things we need to understand and why this takes some time. Like I always, people ask me, I say, boy, I'll tell you, people in the lab are working hard, but there's just a lot of little stuff that needs to get done.
And anything you're expecting that would be unique is a fourth important question from the group. Anticipating special problems or obstacles in humans that you didn't see in mice or guinea pigs. Any unique, say, God, humans are different this way or that way. I'm more worried about this or not.
So there's two. I'll talk about the easier one first. I don't know if it's easier, but one is that AAV, I mentioned, the vector we want to use. It's a natural virus. We take its stuff out and put our transgene in, but we've been infected with the actual virus. All of us.
Immunity to it.
Yeah. So there's a whole bunch of different types. Many of us have pre-existing antibodies to some of those. We've basically been vaccinated. There's the first one, AAV1 or AAV8. They're kind of numbered, and then they get letters and stuff. It doesn't really matter. But, if you've been exposed to one of these in the past, you have this immune response. How effective will the therapy be? And so, one thing we're looking at is having a whole quiver of AAVs. If we can find several that work, then in a given human being, we can find one that they haven't been exposed to in the past. That's the goal. In mice, these mice have other viruses, so you don't have this problem. So any mouse you can put our AAVs into and it works. So we got to figure that out. The other thing that we've been really interested in is, I said that the ganglia. So basically, when you touch your face, around your mouth, anywhere you can feel that. That's because there's nerve endings there. The nerve bodies for those nerves are actually way back up, kind of in the middle of your head or by your spinal cord, and then they just send out these long projections. So we really need to get to those nerve bodies, and that's all the stuff that we really need to achieve. So that's a huge challenge. And what we find is that not all those nerves are the same. So, and different AAVs like to go to different sorts of those nerves, and herpes likes some of those nerve bodies better than others. And so we're trying to separate those out. And that, if you get way in the back of the paper, we did that on a single cell basis. We actually figured out which neurons exactly had herpes and which AAVs like to go there. And then that's all in mice, right? So is that the same in human beings? Well, we started to do that in the guinea pigs, and I'm going to thank the group a couple times and not miss a chance. Those studies started, a year early because of your guys' support. So we're looking at that question in guinea pigs, and it looks like it's the same AAVs work in the guiney pigs too. Now it's a rodent, so that's really cool. But now here's the really cool thing: is I mentioned that I work on other viruses, and in HIV, we had to work. We were involved in some studies that used some rhesus macaques, and we were able to, because they can be infected with HIV. It's one of the few animals that can happen. So we worked with our colleagues who were doing those experiments, and basically piggybacked. We were actually introducing. We made some AVs that they put in. Could we look at the ganglia as well and ask in a primate then, how does this look? And so we'll be able to answer to the same AVs that work in mice, are those exactly the right ones to use in human beings? That's going to be incredibly important, again, as we try to go forward and get this approved, and just know what is likely to work in a human being. But our hope, and we're optimistic that it may be the same AAVs in these different model systems that we've used, and that'll really help pave the way toward a human trial.
And Keith, the last question before we get to the current questions are, how close are we to actually beginning a trial? I know you hate to answer questions like that, but what's the best guess?
So we're just starting now some contact with the regulatory agencies, and just some colleagues that we'll need in order to do these sorts of studies. Big groups. There's rigorous manufacturing that needs to happen for human beings. You have to get the people. I'm shooting for late 2023 is kind of my personal goal. We'll see how that goes. I've been telling people 3 years. You can't do that forever, because I realized that I probably started to say that several months ago, and so we're going to try to get something started in late 2023. But I have to be honest. If we see a safety signal that there's something we need to work through,
right,
that could get pushed back. And I want to be really honest with the folks. I know people are so motivated. People ask me this, and I'll say this again. We're really working as fast, as hard as we can, but we really don't want to hurt anybody. If somebody does get hurt in these, it could push this entire thing back by years. So we need to be really, really careful and move in a very rigorous way.
So one question that's come up, and this is a question, Keith, that I get asked. I must get asked this question every week in different forums and different places. And it comes back with the fact that, we're all delighted with what Larry Corey and Dr. Fauci have been able to create with vaccines for COVID. And my gosh, what an incredibly successful story that we get the sequence in mid-January, and we've got an mRNA vaccine into people by mid-March, which is really unbelievable. And all the vaccines seem to work. Why do vaccines work against COVID? Why do vaccines work against influenza? Why do vaccines work against measles virus? And yet they don't work against HSV?
Yeah, that's a great question. And that's such a logical question to ask. And I always applaud people who come up with that, but haven't spent a lifetime studying this stuff. The So the high level answer, and then I'll dig down a little bit more, but the high level answer is you can think of viral targets for vaccines as easy targets and hard targets. And there are some that are just easy to make vaccines for, and there are some that are inherently hard. So you mentioned some of the easy ones, measles, that's a good one. Smallpox, right? That was where this whole thing got started. Um, Flu, although our flu vaccine is actually not all that, please
Right.
get it, everybody.
Yeah.
Please get it. But we know, in a good year, it's maybe 60 or 70% effective. That's part of why we were so thrilled when the coronavirus was 95% effective. It's just amazing. So there's easy ones and hard ones, and it doesn't correlate with how bad the disease is, right? So smallpox, thank God none of us have ever witnessed this. Huge number of people dying, and this is the one that they basically stumbled into. Farmers stumbled into this, right, with the people milking cows, right, and they found out that they weren't getting it because they were, in their workplace infecting themselves with a related virus, cowpox. And it gave protection against the smallpox. And eventually the scientific enterprise sort of figured this out, and it's led to vaccines. So there are, another great example, that took longer than it should have, if we'd have really put a ton of effort in it, is Ebola virus, right? Horrible, horrible disease, right? 30, 40, 50% of people who get it die of the infection. And yet there's some vaccines, and they actually seem to work pretty well.
Yeah.
And then when the entire world's effort, and literally billions and tens of billions and probably hundreds of billions of dollars get put into things, you can make a vaccine for a global pandemic in less than a year. It's really remarkable. Okay. And then there are viruses where a heck of a lot of effort's been made over a long time, and we don't have vaccines. So those are herpes simplex, that's hepatitis C, that's HIV. Huge amounts of effort have gone into HIV vaccines, and again, the Fred Hutch is the world center for HIV vaccine research. It's all headquartered here. Larry Corey, Julie McElrath, these are globally renowned leaders in this. These are smart people, and I've worked with Larry my entire career. He knows more about this stuff than anybody I know. But frankly, we don't have a vaccine, and it's not that people haven't tried. It's not resources haven't been there. It's not that they're not smart. It's that the viruses are really hard. So how do viruses do this? Some viruses just change so fast that you can't keep up with them, and that's largely what HIV does. It can mutate so quickly. It has so much diversity in it that a vaccine that might work against this particular HIV doesn't work against one in the person next door. It's just, there's so much diversity. You see, that's probably why we don't have a hepatitis C vaccine as well. The other thing is some viruses just have mechanisms of passing from one person to another, or from causing disease, that there's sort of no place for the kind of immunity we know how to generate now by a vaccine to actually ever act. So most of our vaccines make antibodies. That's what seems to work for COVID. But when HSV is transmitting from one person to another, the antibodies just sort of never have enough time to do anything before that virus goes from one person to another, jumps into one of those nerve endings right there in the skin or mucosa, and then it's shielded as it makes its way down to the nerve body, where it establishes this latent infection. Looks like there's just no time for the antibodies to ever actually act to prevent it. And literally, there have been, I would estimate, hundreds of millions of dollars spent working toward an HSV vaccine. Several companies have gone bankrupt because they put all their eggs in that basket and got into human clinical trials, and they just found it just didn't work in the real world. It's very frustrating. Larry's been involved in those for a long time as well. So it's difficult, and so that's why we don't have this vaccine. Recently did have a really nice breakthrough in Varicella Zoster virus. It is a herpes virus. It's the one that causes chickenpox. I'm old enough to have had chickenpox as a child. I got revaccinated now. Chickenpox goes down to very similar nerves. Goes down. We get it. It goes into the nerves, establishes latency. and then it usually truly lies completely dormant for decades. Then it comes back as shingles, which is a really a bad, very painful thing for many people. Don't want to have that. And the vaccine now actually works really, really well. And that took some real insights into not only the vaccine, but sort of all the other components going into something called adjuvants that kind of help the immune system rev up. We found out sort of exactly what parts of the immune system you need to rev up to make that work. Maybe some of that work will ultimately spill over into HSV and maybe help with some HIV vaccines. There are groups working on that. I sure hope that works. That would be wonderful if we could have multiple approaches. That would be a great thing.
Yeah, I actually, my wife and I had we got the vaccine together, the Varicella Zoster vaccine, and we basically spent 2 days watching Netflix after each one because those immune adjuvants were quite potent, and they really got a good immune response.
Tom, I tell people this story that I've been doing science a long time, and I've gone home from work sick once in 25 years of doing this. And it was the day after I got my shingles. I was here in the afternoon in this office. Right. And I said, I can't sit up in this chair anymore. I have to go home. Yeah.
That's what we found.
So for everybody on the line, though, that said, yeah, we felt bad. I went home, I laid down, took an 8-hour nap. That is way better than getting shingles, in my opinion. So I'm not discouraging people. And the other thing I'll be honest with people is I'm hearing that some of those same sorts of reactions are happening with the second dose of your COVID vaccine. That doesn't, some people are getting that. We'll see. I because I work with COVID, I've had 1 dose, not 2 yet. Please get your vaccine. And I'm doing it personally. I believe in these things. I'm putting my money where my mouth is when I say that. I'm getting it. My family will get it when they're eligible. It's way better to deal with a couple hours of Netflix time than potentially fatal illness.
I absolutely agree. So, Keith, we got an issue here. We got 43 questions and we have 12 minutes. So we're gonna try to get through as many.
Lightning round. Okay.
We'll try to get through as many of these as we can. Unfortunately, they're not really easy questions. They're all really hard questions.
It's a hard subject.
So here we go. This is a good question. In an infection with HIV, it's difficult to know which ganglia of the bodies are infected. Do you expect that the AAV, once injected in the bloodstream, will reach ganglia all over the body, or will they get to all the different sites of infection?
Yeah, that's a great question. So we're looking at that. It looks like anatomically, once they get into the body, they can go to ganglia anywhere in the body. There are these differences between ganglia. Some are sensory; they deal with feel. Other ones deal with sort of autonomic issues, like does your pupil expand or contract, things we don't even think about that your nervous system takes care of. Those can have herpes in them too. We think it may take a couple of different AAVs together to cover everything, but we're working on that. It's a very important issue.
And so let me just tell you something I just did. I'm very proud of this one. The question was asked in Spanish. I don't speak Spanish, but I went to Google Translate really quickly and was able to get it written in English for me. So shows you how good Translate is. And this one is, Dr. Jerome, if approved, will this be on sale globally in all countries? And what's the estimated time for it to reach other countries once you demonstrate that it works?
Yes. So the absolute goal for anything that I do is it's available worldwide, and we're especially, I'm not sure where you're writing from, but a Spanish-speaking country somewhere, I assume. We're very tuned into making our cures available for low resource areas of the world. We think about that particularly for the HIV work, where the major burden is in areas that just don't have the resources we do, and we understand that. So we try to make sure that everything from day one, we're thinking about how do you make this cheap enough it can go everywhere, everybody who needs it can get it. So yeah, we'll do that. The reality is there are regulatory issues as you try to go in other parts of the world. We will make every effort to deal with those. And frankly, we will need to work with others who have dealt with those sorts of issues in the past and know how to work with all parts of the world. But But the short answer is yes, we're going to get it to every part of the world.
And next question, Keith. I know I get asked this question about cancer all the time, and I'm sure you get asked this question about HSV and other viruses, which is how long are these trials going to take? And, will this be the case where you're going to have to go through phase 1, phase 2, phase 3, and it could last 7 to 10 years to get there? How long is it going to take?
Well, I'm sure the answer is that we will not be allowed to cut any corners. So there will have to be phase 1, phase 2, and phase 3. There's just it's not going to be like Operation Warp Speed in COVID. Not the corners were cut, but everything was compressed, kind of almost done, back to back. And the reviews from the government happened, the day that the data got in, and these sorts of things that are just, it's not going to happen here, and it's just the reality, and it's not because I don't want them to, but we're going to need to be extremely careful again because we can't hurt anyone with this. So there'll be phase 1, phase 2, phase 3. Will it take 7 to 10 years? I don't know. When I say trial in 2023, that'd be a phase 1 clinical trial, small group of people, 20, 30 participants probably, just essentially looking that it's at the first glance it seems to look safe in those people. And then we'll obviously look for signs that it worked or not in those folks. But the phase 1 trials are really just about it being safe. And then you go to the phase 2, you open up a couple hundred people maybe. Again, you're looking to make sure it's safe. Now you get a little better handle on is it really working or not. And then phase 3 is the real, the money experiment that leads to approval. And that's typically thousands of people. You heard the vaccine studies for COVID. Each one had typically about 30,000 people in them. Over months, and we'll talk with the regulatory agencies. I don't know that we'll be demanded to have 30,000 people in the studies, but it will probably be numbered in the thousands.
Yep. Next question is a good one because I get this question I'm a medical oncologist, so I get this question as a cancer doctor a lot. And this is a question that says, how can we better educate physicians in the infectious disease field to keep up with cutting-edge HSV research? My experience with these MDs has been terrible. They seem to know almost nothing beyond Valtrex, which doesn't work for all. How do you answer that question?
Yeah, I'm not sure I have a good answer for that, frankly. I could personally had those sorts of experiences in healthcare system, as well in terms of, variety of health issues and providers and, and as a scientist myself, I'm able to do some direct educating of folks. The fact that you guys are on this call, clearly, you're informed, you're involved, you're advocating. That's so powerful. I've just really been thrilled to see that in this community, and I think it's going to be a grassroots effort. I'm really sorry to say that. I do everything I can. We try to get the word out. We try to do opinion pieces and talk. I give seminars all the time. I talk to doctors, but I'm not going to reach everybody, and especially those. You got one who's not really going out there trying to learn. What are you going to do there other than take that opportunity? Try to think of the next person who's in a similar situation that you are. The education you do may benefit someone else. Let's all pay it forward, and I think we can make some progress.
And again, this shows you how well informed this audience is. This next question says, I remember you mentioned in a previous lecture that AAV effectiveness was different in the trigeminal ganglia versus the sacral ganglia. Is this still the case, or did I interpret that correctly?
No, you definitely got it right. So the trigeminal ganglion and the dorsal root ganglia. So the trigeminal ganglia is the one in the head, so the innervates the mouth and face. The dorsal root ganglia are along the spinal cords where they innervate the rest of the body. Those are pretty similar. They're sensory ganglia. If we look, their neurons seem pretty similar. The AAVs that work for those are pretty much the same, I think. Then there are other ones like the superior cervical ganglia. That's one of these autonomic ganglia I was talking about. There's a number of those that seem to be involved. They're sites that the virus can reactivate from and cause recurrences. So we need to cover those as well. and that's part of the reason to try to get those two quite distinct types of ganglia. That's part of the reason why, at least at this point, we're needing to use combinations of AV to get full coverage. In the paper, we used combinations of 3. If you look carefully, the third one didn't add a whole lot. It really seemed like it was the 2 that are important to make sense, because there are these 2 big groups of ganglia we need to cover that are both seem to be important. Yeah, so we're working on that. I think your interpretation is correct, and I think we've largely got it covered, but we're still looking.
Here's a great this comment. It's not a question, it's a comment. It's gotten a ton of likes.
Okay.
And it's from someone named Tom Andrus, and it says, Dr. Martine Albert has led much of the work in the lab. Thank you for your hard work, Martine. So I think we want to thank.
I'm gonna like that, because Martine's been working with me for almost my entire working career. Not quite, but she was one of the very first people that I hired, and she's amazing and so important to this work, and knows more about HSV than anybody I know. So yeah, she should get an incredible amount of credit for this. I'm privileged to work with her.
And one thing I'm really struck by, just looking through the chat line, is how much international support there is. There's somebody from Norway, somebody from Brazil, somebody from one of the Spanish-speaking countries. Not sure, it could be the United States. Not sure which one. But it's great to see the international support for what you're saying, which is what you're doing, which is terrific.
These viruses don't know any borders, Tom. The borders are all artificial, and we're, it shows our humanity, right? And our shared humanity, and the fact these viruses don't care in the least if you're in Norway or you're in Colombia. It doesn't matter.
Yeah, no, that's absolutely true. And there's a question here about nerve damage from the virus, and that is a question as, Dear Dr. Jerome, thank you so much for this. I'd like to ask about nerve damage. Is it normal with HSV-2? My doctor thinks I'm overreacting, but I know prior to acquiring this disease, I never felt these sensations. The sensations are pins and needles, burning, and sharp shooting pain in the legs. Could that be from HSV, or could it be just the neuropathy that's pretty common in a lot of people as they age?
Yeah, well, sort of either one of those are possible, and it's very, very hard to separate those out, even working directly with people. And I have clinical colleagues that I talk about with these sorts of things at the University of Washington. So it's hard to say, and I certainly couldn't really comment on this particular person from a distance. This is really not my area anyway, but it certainly can happen. And that's one of the things that we're really looking at. When I say safety, one of the things of our therapy, we want to make sure that that the therapy doesn't cause any nerve damage as well, right? I mean, there's a number of things we think about. We need to make sure that this is safe, and those things could happen. And the last thing we want to do is make them worse.
Great. So I want to thank everyone who's participated today. We just got a couple of texts from Canada, a couple of texts from Spain. So again, thank you very much, and really appreciate everything. I want to thank Keith for everything he's done, and more importantly, thank all of you for what you've done to support Keith's research. There is no doubt that this is an area that's not on the top priority list of all the pharmaceutical companies in the world, and often given priorities of other diseases, it needs a motivated base like the group who's on this call to help keep Keith's lab going as aggressively and moving forward as much as they can. And I really want to again thank you for everything you're all doing. I hope today proved to be a great opportunity to learn about what's happening with Keith's work. I, for one, learned a ton about HSV and the work that he's doing, and I really think that it's great that you're all here. And Keith, as always, I love doing these Q&As with you, and look forward to the next one. Thanks, folks.
Oh yeah.
Bye bye.
Take care. Thanks, everybody.